Hello students, this is Lohita. Here we are into zoology NEET class, and today we are going to learn the continuation concepts related to molecular basis of inheritance. Okay, so today we will be learning about adapter RNA, okay, which is nothing but tRNA. See students, right from the beginning of a proposition of the genetic code, Francis Crick, okay, right from the beginning of proposition of genetic code, Francis Crick, he very strongly believed, okay, right from the proposition of genetic code, Francis Crick, he very strongly believed that there must be something to read the genetic code and to add amino acid to the proteins, okay, because see these amino acids, they have no structural complementarity with the RNA. Amino acids, they have no structural complementarity, with the RNA, isn't it? No, they have no structural complementarity with the genetic code. There is no structural complementarity on the genetic code, right? No complementarity is found between the amino acids and the genetic code which is present on the RNA. So then how can the amino acids be added to the polypeptide chains? This was a very big question. What is adding these amino acids to the polypeptide chains? Okay, so there Francis Crick has proposed the presence of a soluble RNA. Okay, Francis Crick has proposed the presence of soluble RNA, and this concept of soluble RNA was proposed very much before the proposition of genetic code. Very much earlier to the proposition of genetic code only, the Francis Crick, he proposed the presence of a soluble RNA. This soluble RNA is actually reading the genetic code and bringing the amino acids according to the genetic code and adding it to the polypeptide chain. Okay, so this soluble RNA is reading the genetic code. It is reading the genetic code and bringing the amino acids. It is reading the genetic code and bringing the amino acids and adding it to polypeptide chains. Okay, so he believed that there is the presence of a soluble RNA which is actually reading the genetic code and bringing amino acids and adding it to the polypeptide chains to form proteins. Okay, so that was a soluble RNA. Later on, this soluble RNA was identified to be tRNA. Okay, so this tRNA is actually transfer RNA. Transfer RNA is the one which reads the genetic code, and this brings the amino acids according to the genetic code. It reads the genetic code on the RNA and it brings the amino acids according to the genetic code that it has read. Okay, according to the genetic code, it is also bringing the amino acids. Okay, so that is the importance of tRNA. What is this tRNA consisting of? How is it able to read the genetic code? How is it able to bring the amino acids? Okay, these were all the very big questions. Then later on, the structure of tRNA has been identified. You know, this tRNA appears like this. Okay, so it has one anticodon region. And it has one acceptor arm. Okay, it has an anticodon region and it has an acceptor arm. Okay, onto the anticodon region, this tRNA will have the nucleotides which are complementary to the mRNA. Anticodon region, the tRNA is having two regions, as I have told you. The first region is anticodon region. Okay, anticodon region will have the codons anticodon will have the nucleotides which are complementary to the nucleotides on RNA, which are complementary to the nucleotides on the mRNA. This is the anticodon region. Anticodon region of the tRNA will contain nucleotides which are complementary to the mRNA. It will contain the nucleotides which are complementary to the mRNA. Okay, and on the other side it is having an acceptor arm. To this acceptor arm, the amino acid according to the anticodon will bind. Amino acid will be present on the acceptor arm. Amino acid will be present. Okay. So in this way, the tRNA will have an acceptor codon and an acceptor arm and an anticodon. Okay. Let us take a look at the example for a tRNA coding for an amino acid. Okay. So, for example, anticodon is consisting of AUG. This is the codon and this is the nucleotides on anticodon. Okay, anticodon arm. This is the anticodon arm. Okay, on the anticodon arm, the tRNA is having AUG. And this is the mRNA, students. This will be the mRNA. On the mRNA, the anticodon will be having its complementary nucleotides. UAGC. This will be the code, this will be the nucleotides on the mRNA. So, according to these nucleotides, it is having the anticodon and it is carrying the respective nucleotide. Okay, AUG codon will be coding for amino acid tyrosine. So, it is carrying the tyrosine amino acid on the acceptor arm. It is carrying the amino acid tyrosine on the acceptor arm. So, in this way this tRNA is binding with the anticodons on one side and onto the acceptor arm it is carrying the amino acid tyrosine. Okay. So, according to the anticodon it is carrying the respective amino acids. So, it reads the mRNA's codons and it is able to, the tRNA will be able to bring the appropriate anticodons and amino acids onto the polypeptide chains. Okay. So, you know students, there is every mRNA which is translated into proteins is beginning with AUG, isn't it? AUG is the initiator codon. AUG is the initiator codon. Any mRNA which is being translated into proteins will be beginning with a codon AUG. This is the initiator codon. Right. So, because initiator codon is common to all the proteins will be beginning with the amino acid methionine which is coded by AUG, right. This is the initiator codon which is coding the amino acid methionine. Every polypeptide chain will be beginning with methionine which is coded by the initiator codon AUG. Okay. So, students, because AUG is common to all the amino acids, this is initiator codon. So, initiator tRNA is also present, okay, for coding this amino acid methionine. There is in initiator tRNA. Okay. So, initiator tRNA is responsible for binding the binding of methionine amino acid. Initiator tRNA will bind with that methionine amino acid and it will bring the methionine amino acid. Okay. Initiator tRNA will be bringing the methionine amino acid because every polypeptide chain starts with methionine. So, every translation will begin with the initiator tRNA. Okay. So, but students, there are stop codons are there, they are not coding for any amino acids, right. Stop codons are there, they are not coding for any amino acids. Because the stop codons are not coding any amino acids, there are no stop tRNAs, no tRNAs are there for the stop codons. Stop codons like UAA, UAG, UGA. These are the 3 stop codons, right? So these 3 stop codons, they are not coding for any amino acids. They don't code for any amino acids. They are not coding for any amino acids. So there are no t-RNAs for these stop codons. There are no t-RNAs for the stop codons. Okay, there are no t-RNAs for the stop codons. Because there are no t-RNAs for the stop codons only, that translation is stopping when it is reaching the stop codons. Okay, so there is no t-RNAs for the stop codons. Okay, students. So that's all related to t-RNA or adapter RNA. We have learned that Francis Crick proposed this idea of presence of t-RNA. They were earlier known as soluble RNA, but now later on they were called transfer RNAs, because they are transferring the amino acids onto the polypeptide chains. They are reading the codon on the mRNA. They are having the anticodon for that codon on mRNA, and they are also binding with an amino acid onto their acceptor arm, adding the amino acids to the polypeptide chains. They are called transfer RNAs. Stop codons are not having any transfer RNAs. Start codon is having the same RNA, that is initiator RNA. Okay, so that's all about adapter or transfer RNAs. Okay. Then we will learn now about translation. We are now going to learn about what is the mechanism behind translation. Okay. So what is translation, students? Translation is nothing but adding of amino acids together, adding of amino acids together, that is polymerization of amino acids. Polymerization of amino acids to form a polypeptide. To form a polypeptide is called as translation. Adding of amino acids together, that is polymerization of amino acids to form a polypeptide is called as translation. Okay. So this translation is nothing but amino acids are formed, they are making up the polypeptide chains, that is proteins are formed from mRNA. Okay. From mRNA proteins are formed, that is called as translation. So based on the sequences of nucleotides on mRNA, based on the sequences of nucleotides on mRNA only proteins are formed. Okay, how the proteins are formed? Based on the sequence of nucleotides, based on the sequence of nucleotides on mRNA only proteins are formed. Okay. So tRNA is very crucial in the transfer of amino acids onto polypeptide chains. tRNA is very crucial in the transfer of amino acids onto the polypeptide chains. Okay. So here students, the amino acids are linked to each other. Amino acids are linked to each other by peptide bonds. Okay, peptide bonds will be connecting the amino acids to each other. So peptide bonds will link the amino acids to form polypeptide chains, to form polypeptide chains. Okay, to form polypeptide chains, the peptide bonds are formed between the amino acids. Okay, and peptide bonds require energy for formation. Energy is required for the formation of peptide bonds. Energy is required for the formation of peptide bonds. Okay. So initially only before the amino acid is transferred onto the polypeptide chains, only the adding of energy will be occurring. Okay. So before polymerization of amino acid chains only, the transfer of energy onto the tRNA will occur. Okay. By utilizing energy, amino acids are transferred onto the tRNAs. Okay. Amino acids are transferred onto the tRNAs by utilizing energy. Such tRNAs which are carrying the amino acids are called as charged tRNAs. They are called charged tRNAs. They are called as charged tRNAs. That is called as charging of tRNAs. That is called charging of tRNAs. Okay, that is called charging of tRNAs or amino acylation of tRNAs. Amino acylation of tRNAs. Okay, amino acylation or charging of tRNAs is nothing but adding of amino acids to the tRNA. That is called charging of tRNAs. This is an energy expensive process. This require energy. Energy is spent during this process of binding of amino acids to the RNAs. Okay, so this is the process. This is the time where energy is spent during the formation of amino acids. Okay. So, now this charged tRNAs, two charged tRNAs will come closer, okay. Two charged tRNAs will be coming closer to each other and a peptide bond will be forming between these two charged tRNAs, okay. Two charged tRNAs will come closer and peptide bond is formed between them. Peptide bond is formed between them. Okay, when two charged tRNAs are coming closer to each other, then the peptide bond is formed between them. Two charged tRNAs will be coming close to each other, then a peptide bond will be formed between them, resulting in the formation of amino acid peptide bond. So, now when two amino acids are binded by peptide bond, it will form dipeptide. 3R bond tripeptide. In that way, many amino acids will be linked up to each other, forming a polypeptide. A polypeptide is nothing but a protein. Okay. See students, yes, we have studied that translation is occurring by utilizing RNA as a template strand. Complementary to the RNA, tRNAs are having anticodons and they are adding the amino acids to the polypeptide chains, right? Very good. Now, where are these amino acids, where are these translation occurring, where are these proteins formed? Ribosomes are the sites for protein synthesis, students. Ribosomes are the sites for translation. Ribosomes are the main sites for translation. Ribosomes are the main sites for translation. Translation of amino acids will occur in the ribosomes itself. Okay, the ribosomes, they contain two subunits. Okay, ribosomes contain two subunits. Okay, and they contain they are acting like a catalyst. They are acting like catalyst for translation. They are acting like catalyst for translation. They contain nearly 80 different type of proteins present in them. Okay, they contain nearly 80 different types of proteins present in them. Okay, so the ribosomes are having two subunits, right? So those two subunits of the ribosome will be, the two subunits of the ribosome would be one larger subunit and one smaller subunit One subunit will be a larger subunit, one subunit will be a smaller subunit. Okay. So, let us see how these are involved in forming of a polypeptide chain. Okay. See students, yes, so this is the small subunit for suppose. This is the large subunit and this is the mRNA. Okay, this is the mRNA. So, the smaller subunit, you see, the smaller subunit is binding with the mRNA. What is the importance of smaller subunit? The smaller subunit binds with the mRNA. Okay, and the larger subunit, it always contains two sites for binding of tRNA, It always carries two sites for the binding of tRNA. Okay, it is always carrying two sites for binding of tRNA. Okay. So, students, now let us see how it is forming. I will just show you with an example. This is the t-RNA, this is another t-RNA. Right. So, students, there are two binding sites for tRNAs, and this will be the mRNA, right. Let us write even the code on mRNA. Okay. Right. So, students, there are two binding sites for RNA in the larger subunit. Okay. In the larger subunit, there will be two binding sites for RNAs. Okay. So, now students, on the smaller subunit tRNA and the smaller subunit mRNA will be binding. Okay, on the smaller subunit mRNA will be binding. Now, the tRNAs will be coming according to the codon present on the mRNA. Okay, the smaller subunits will be now added according to the codons which are present on the mRNA. See, what is the codon in mRNA? UAC. So, this codon is bringing AUG. Okay, this is not methionine, this is tyrosine. Okay, so AUG. Okay, so AUG is a anticodon for UAC. So, this tRNA is having AUG anticodon on it. It has come and bound with the first site on the first binding site on the larger subunit. This is the first binding site, it has bounded. Now, onto the second binding site, there is anticodon CGU, which is going to carry amino acid alanine. So, now between this tyrosine and alanine, which are placed side by side, a peptide bond will be forming. And after that, this tRNA will slide away, this tRNA will go outside, this will be released from tyrosine, and after that this alanine will come back to its second position, it will come back to first position. Okay. So, now what is happening, students, here? Two binding sites are there, two different RNAs are binding with two different tRNAs are binding with the two binding sites. Now, between first and second binding site tRNAs, a peptide bond is formed. After peptide bond is formed, the first site amino acid is joined to the polypeptide chain. This tRNA will be leaving the amino acid. This second amino acid will be moving to second tRNA will be moving towards the first position. Now, on to the next site, and fresh tRNA will be coming. Okay, this will be moving away, this will come to the first position, a new tRNA will be coming, and this new tRNA will be binding to the second binding site, and then a peptide bond will be formed between the second and third amino acids. Okay, in that way, when the amino acids are brought onto the binding sites of the larger polypeptide, at that time only the formation of peptide bonds are occurring between the amino acids. When the peptide bonds are formed between amino acids, when they are binding the first and second when they are present on the first and second sites of the larger subunit, at that time only the formation of peptide bonds between the first and second amino acids is occurring. Okay, so this is how the peptide bonds are formed, students. So, and you know, the peptide bonds are formed this way continuously, new peptide bonds will be forming, and finally when the stop codon, when the ribosomes are reaching the stop codon, the stopping of the translation will occur. When the ribosomes are reaching the stop codon, stopping of the translation will be occurring. Okay. So, students, here continuously right from the initiator codon till the terminator codon, the ribosomes will be moving from one codon to another codon. Okay. The ribosomes will be moving from one codon to another codon, one after the other they will be continuously moving and coding for the proteins in continuous manner till they reach the stop codon. Till they reach the stop codon, this moving of our ribosomes one after the other codon will be continuing. Okay. So, students, with the initiator codon, the translation will start. With the initiator codon, the translation will start, and with the stop codons, the translation will be stopping. Okay. With initiator codon, translation will start. With the stop codon, translation will be stopping, because stop codons are not coding for any amino acids. When the mRNA is reaching the stop codon, then then the translation is going to stop. Okay. So this is how stopping of translation occurs, students. So between 3 and 5 dash ends, between the 3 and 5 dash ends, the translation will be continuing. Okay. And then this is occurring with the help of tRNAs. Okay, ribosomes are acting like catalyst for translation. tRNA is acting as an adapter. It is bringing the required amino acids and adding to the polypeptide chains. Okay. And you know, students, in the mRNA there are some untranslated regions also. Untranslated regions are also present on the mRNA. On the mRNA there are some untranslated regions also. Untranslated regions in the sense what? Untranslated regions are those regions of the mRNA which are not translated. Okay, these are the regions of the mRNA which are not translated. Translated regions of the mRNA exist between the initiator codon and terminator codon. Between the initiator codon, and terminator codon, between the initiator codon and terminator codon is the translated region, So beyond that, before and after the initiator and terminator codons, there are untranslated regions on mRNA. The untranslated regions UTR, short form UTR, which are present on the 5 end, also are called 5 end untranslated regions, 5 end untranslated regions. And the untranslated regions present on the 3 dash end, they are called as 3 end untranslated regions. Okay. So what is the importance of having these untranslated regions? The main importance of having these untranslated regions is, these untranslated regions are important for translation. They will protect the mRNA from damages. They will ensure that translation is occurring between the regions necessary only. Okay. So translation of the required regions is enabled because of the presence of untranslated regions. So untranslated regions are also important for the effective translation to occur. Okay. So untranslated regions are also important for translation to occur. So these are few concepts related to translation, students. So students, then let us learn few more points related to translation. Okay. So students, translation will be occurring between initiator and terminator codons. Okay, between initiator and terminator codons, between initiator and terminator codons only translation will be occurring. Okay, translation occurs between initiator and terminator codons only. So here the mRNA will be read by the small subunit of the ribosomes, right. So this mRNA, this ribosomes will be passing from one codon to another codon in that way. Okay, and with the initiator tRNA binding with the initiator tRNA onto the larger subunit, binding of initiator tRNA onto the larger subunit, translation will begin. Okay, translation begins. And now when the initiator tRNA, after initiator tRNA, the ribosomes will move from one codon to another codon continuously till they reach the stop codons, Okay. So when they are reaching the stop codons, there is no tRNA for which is coding for stop codons. So that is why what happens here is a release factor will bind with the mRNA, okay, which results the release of polypeptide, which causes the release of polypeptide. Okay, So stop codons are not coding for any amino acids, then what happens? The release factor will be binding with the mRNA, and binding of the release factor to the mRNA will make the polypeptide to be released from the ribosomes. In that way this polypeptide is free now, it can go and do its work. Okay. So this is how the translation is occurring, students. That is all about translation. Now we have to learn about the next concept, that is regulation of gene expression. Regulation of gene expression. See students, genes, they are expressed as RNAs, mRNA particularly. mRNA will be expressed as proteins, right. So genes are expressed as mRNA, mRNA will be expressed as proteins. So here students, for regulation of gene expression can be done at any place. Regulation of gene expression can be done in the transcription of mRNA, or in the processing of mRNA, or in the formation of proteins. At many different levels, according to the requirement of the activity, the gene expression can be regulated at many different levels. According to the requirement of that particular activity, gene expression can be regulated at many different levels. Okay. So gene expression can be regulated at 4 different levels. Can be regulated at 4 different levels. Okay. They are, the first one is during transcription of mRNA. During transcription of mRNA. You see students, in case of organisms which are containing DNA as a genetic material, in the organisms which are containing DNA as genetic material, DNA is not causing any expression of the characters, isn't it? DNA is never going to cause any expression of the characters. DNA will be expressed in mRNA or a tRNA or rRNA, and it is causing its expression. So DNA is now dependent on RNA for its expression. Some of these RNAs are all involved in protein synthesis ultimately, whatever is the type of RNA involved in protein synthesis ultimately. Okay. So students, in our organisms containing DNA as a genetic material, gene expression is regulated through the formation of RNAs, or by the formation of proteins from the RNAs. Okay. So we are completely dependent on the RNAs for the expression of gene, directly or indirectly dependent on RNA for the expression of genes. So gene expression is regulated at transcription level during transcription of RNAs. Any RNA, during transcription of RNAs, the gene expression can be regulated. Okay. And the second way is during splicing of RNA. During splicing of RNA. See students, our RNA is not fully functional when it is produced. Non-functional RNA is produced. Primary transcript, we call it as primary transcript. We call it as primary transcript. That RNA which is not fully functional, is called as primary transcript. Okay. So this primary transcript is the one which is undergoing some processing to form a functional RNA. So this is not functional. Primary transcript is not functional. It is just an initially formed RNA. Okay. So this in primary transcript will be undergoing a process called splicing, where the non-expressed region introns, they are removed. Unexpressed regions, introns are removed from the RNA. Okay. So according to the type of introns removed also, the expression of genes is regulated. This is particularly in case of eukaryotes. This is particularly in case of eukaryotes. Okay. This is very particularly in case of eukaryotes, because splicing of RNA is seen only in eukaryotes. In prokaryotes there is no splicing of RNA seen. Splicing of RNA is seen only in eukaryotes. So eukaryotes only regulation of gene expression is also through splicing of RNA. Okay. And the next method of controlling gene expression is formation of transport of RNA, transport of RNA transport of RNA into the nucleus, during transport of RNA into the cytoplasm, from the nucleus. See students, RNA is formed inside the nucleus. This RNA has to be safely transported into the cytoplasm to show its effect. So safe transport of RNA into the cytoplasm is also very important. Here also gene expression is regulated. Okay. And the next step is translation. This is a very important step. Translation of RNA to form proteins. This is another crucial step which is occurring in the gene expression. Okay, so these are the 4 ways by which 4 levels where gene expression is regulated: during transcription of RNA, splicing of RNA, transport of RNA into cytoplasm, translation of RNA into proteins. These are the 4 steps where the gene expression is regulated. These are the 4 levels where the gene expression is regulated. Okay students, so that's all how the gene expression is regulated students. You know, in case of prokaryotes, as we have already studied, in case of prokaryotes, transcription and translation occur simultaneously. Transcription and translation, these both events occur simultaneously. Okay, both the events, transcription and translation, these 2 events occur simultaneously in the prokaryotes. Okay, so that means in case of prokaryotes there is very particular regulation of gene expression. Gene expression is in these animals mainly regulated during transcription level, because transcription is occurring along with the translation. Whatever mRNA is transcripted, and then it is at the same time undergoing translation. So transcription and translation are occurring simultaneously in prokaryotes. So you know, some of the factors whatever we have discussed here are only applicable to prokaryotes. That is transcription of RNA and translation of RNA into proteins. Only these 2 levels of gene expression, regulation of gene expression is applied to prokaryotes, because transcription and translation are occurring simultaneously. Transcription is gene expression is regulated at transcription level, translation level. We do not observe any splicing or transportation of RNA into the cytoplasm in case of prokaryotes. No splicing is occurring, no transport of RNA is occurring. That is why these 2 are not the factors which control the gene expression in case of prokaryotes. Okay, so students, the gene expression of prokaryotes is regulated in a different manner to that of eukaryotes. In prokaryotes, gene expression is regulated by binding of RNA polymerase, to a promoter or to an inhibitor. Okay, binding of RNA polymerase to a promoter or inhibitor, depending upon that factor gene expression is regulated in prokaryotes. If the RNA polymerase is binding to a promoter or inhibitor, depending upon that RNA transcription and translation are regulated in prokaryotes. Okay, so if the promoter is binding, if the RNA polymerase is binding to the promoter, then gene expression occurs. If the polymerase is binding to the inhibitor, then there is no gene expression. Okay, in that way gene expression is regulated by binding of the polymerase to the promoter or to the inhibitor sequence of the RNA. By binding with the promoter or inhibitor sequence on the RNA, expression or non-expression of the genes is occurring in case of prokaryotes. Okay, so there is one very clear model proposed to explain the gene expression in case of prokaryotes, that is called as Lac-Operon model. Lac-Operon model is proposed to explain the regulation of gene expression, which is very clearly explaining us how gene expression is regulated in case of prokaryotes. This concept we will be discussing in tomorrow, students. Along with a very clear description of Lac-Operon, we will be learning this concept in our tomorrow's class. So here in today's class we have learnt about t-RNA, we have learnt about translation, regulation of gene expression at different levels, in prokaryotes at what levels, in eukaryotes at what levels, we have learnt in today's class. Okay, so that's all for today, students. We will continue to learn more. Few concepts are left over in molecular basis of inheritance. We will be done with molecular basis of inheritance in this week. So that's all for today. We will meet again in the next class. Thank you all for joining the class.